Hello, everybody, and welcome back to biosc140 Human Physiology. This is the part 2 video for membrane dynamics. Remember, this is on lecture exam 1. So part 1, we talked about the membrane itself. Right now, in this part, we're going to talk about movement across that membrane. So membranes in your body form many compartments. Each one of your cells is its own little compartment. And within that compartment, there's smaller compartments, nuclei and lysosomes and mitochondria. And then those compartments that are cells are contained in larger compartments like tissues and separated by other tissue membranes, not cell membranes, tissue membranes. So there's many compartments in your body, and this allows for unique environments within those compartments, which allows for unique chemical reactions to take place within those specific environments. Now, our bodies have to move things into and out of these compartments. And so we're going to look at the movement of things into and out of those phospholipid bilayers, those cell membranes. So there's two big categories. There's active and passive. And what these refer to is the use of energy. Now there's subclasses of each of these types, which we'll get into. But the first big division when we talk about something moving across a membrane is, does that movement require energy, or does that movement not require energy? If it does not require energy, it is passive. No energy input required. Energy input required. Particles move down a gradient. So I'm going to talk about things moving down a gradient, which is with a gradient, or up a gradient, against a gradient, throughout this class. And what that means is if something moves down a gradient, it's moving from high to low pressure or concentration. It's moving from high to low. It does not require any additional energy input in order for the movement to happen. That gradient, that pressure gradient or concentration gradient, is already set up. It's going to move from high concentration to low concentration. Particles move down a gradient. So that gradient has all the energy already in it required for things to move from high concentration to low concentration. No energy input, no ATP used. With active, energy input is required. So ATP is needed to be used in order for that movement across the membrane to happen. Particle is going to move against a gradient. So this means low concentration to high concentration, or low pressure to high pressure. Low concentration to high concentration is against the gradient. And you need to use ATP in order for that to happen. All right, so let's jump in and let's look at the 5 subtypes of passive transport. So all these types of transport right here do not require the use of ATP. They do not require the use of ATP. No energy input. In all of these situations, particles are going to move down a gradient from high concentration to low concentration. And particles are going to move from high concentration to low concentration in all 5 of these manners until there is a state of dynamic equilibrium. Until there is a state of dynamic equilibrium, or equalized concentration on both sides. Equalized concentration on both sides. Now, if you have equal concentrations on both sides of a membrane, there's no concentration gradient. So it kind of makes sense. The energy required for passive transport comes from that concentration gradient. That concentration gradient is the energy needed for passive movement. So if there's no concentration gradient, concentrations are equalized, you're at equilibrium, there's going to be no net movement. But Before we really jump into each of these types, I want to point out this dynamic equilibrium term right here, dynamic equilibrium. So if we look at this portion of the slide, you can see that there's even concentrations of blue hexagons on either side of the membrane, equal concentrations of blue hexagons on either side of the membrane. Now, there's going to be no net movement. So there's not going to be a time where a bunch of these blue hexagons from the bottom side move to the top side and the concentrations are different. But these blue hexagons can still move through this membrane. But for every one blue hexagon that moves up across the membrane, there's going to be one that moves down. So it's going to stay in equilibrium, but it's going to be a dynamic equilibrium, a moving equilibrium, a changing equilibrium. So one blue hexagon goes up, one blue hexagon goes down. There's going to be an equal dynamic equilibrium. All right, let's jump into it. So our first one is simple diffusion. Simple diffusion is defined as net movement of like particles from an area of high concentration to low concentration. Net movement of like particles from an area of high concentration to low concentration. So our example is going to be, one other thing I want to point out. There's no movement across a membrane in this. This is movement through, like within a compartment. This is movement within a compartment. There's no membrane really described right here with simple diffusion. It's movement within a compartment. And let's look at our example. We've got a cup of coffee and we drop a sugar cube into it. What happens when you drop a sugar cube into your cup of coffee? Well, that sugar cube is going to start to dissolve. Right next to that sugar cube, those dissolved particles are going to have a high constant, that area right around the sugar cube. As those molecules dissolve, you're going to have a high concentration of sugar molecules dissolved in your coffee. And those molecules are going to bounce around, going to bounce around, going to bounce around. And as they bounce around, they're going to slowly move away from that sugar cube. They're going to start to move throughout your coffee cup. Now, as that sugar cube is still in your coffee and as more particles are becoming, dissolving, you're going to have a high concentration next to the cube, and those particles are going to be slowly drifting away. And the further away you get, the lower the concentration of those sugar molecules, those glucose molecules. And this process is going to continue and continue and continue until that sugar cube is 100% dissolved. And once that sugar cube is 100% dissolved, you're going to keep having these sugar molecules move in this direction. They're going to continue to move this direction. They're going to continue to move this direction until there is an equilibrium, until you look at image B, until there's an equilibrium throughout the chamber. Throughout the chamber, you're going to have even concentration throughout your coffee cup, even concentration throughout the chamber. Now, are these particles still moving? Absolutely. All of these sugar molecules are bouncing around all over the place. Over time, this particle right here might bounce around and end up over here. Over time, this particle right here might bounce around and end up over here. They're always moving, but they're also always at an equilibrium, even concentration. There's a dynamic equilibrium. Concentration is equalized throughout the chamber, but particles continue to move. The rate at which it achieves this dynamic equilibrium is influenced by temperature, energy input, so that would be using a spoon to stir your coffee, and also molecular size. The larger the molecule, the slower it's going to achieve it. Now we're looking at the next one. The next one is, the next four are all different forms of movement across a membrane. So simple diffusion, no membrane. We're now going to look at ones with a membrane. So direct diffusion, direct membrane diffusion, direct diffusion across a membrane. So this is net movement of like particles across a membrane from areas of high concentration to low concentration. So in part one, when we talked about phospholipid bilayers, the cell membrane, we said that the smaller a molecule is, the more lipophilic or lipid-loving a molecule is, the better it can move across the cell membrane. So molecules like urea can move really easily across the membrane. Glucose is large and lipophobic. It does not move very easily across the cell membrane. So net movement of light particles across a membrane from areas of high concentration to low concentration. If we look at our image up here, we have time. So the more to the right we go, the longer the time. Right here, we have lots of blue hexagons outside of the cell membrane. And down here, we have no blue hexagons. Now, these molecules are going to diffuse. They're going to slip past the membrane. And they're going to move past the membrane and start to build up the concentration below the membrane while decreasing concentration outside the membrane. Eventually, after enough time has passed, we're going to have an equilibrium where both sides have equal concentrations of blue hexagons. Now, dynamic equilibrium, there's still going to be blue hexagons moving up, just like there's still blue hexagons moving down, but the rate of the particle movement in both directions is going to be the same. So how long is it going to take to reach this dynamic equilibrium? How long is it going to take to go from all the blue hexagons right here to equal hexagons on both sides? Well, the rate at which the blue hexagons move across the membrane is called the flux. It's the speed at which the material is traveling across the membrane and appearing on the other side. Now, not the speed of one individual one traveling, but the speed at which these particles as a whole move across. And flux is influenced by 4 variables. Once you know these 4 variables, I'm not going to say in this class I'm going to ask for, but in every semester that I have taught, I have asked many questions on the test about these 4 variables and about direct diffusion, direct membrane diffusion. So let's look into these 4 variables. So concentration gradient is directly proportional to flux. So concentration gradient right here. Concentration gradient is one of the variables that affects flux or affects the speed at which these particles move across the membrane. So it's shortened to Cg, concentration gradient. So if you look at these 2 chambers, let's compare A to B. Which chamber is going to have a greater flux across this membrane in the middle of the chamber? Well, let's think about what we know here. Membranes are identical. The only thing that's really different in this is the concentration. There's lots of red circles up here. There's only a few blue circles up here. The red circles are going to move more rapidly across this membrane than the blue circles. Diffusible particles will reach a dynamic equilibrium if the net flux is 0. All right. Now let's look at this example. Now in this example, we have the same concentration of red circles and blue circles in the upper chamber at the initial onset, but the membranes are different. You only have 3 slits. You only have a small membrane right here, a small surface area of the membrane here, and you have a large surface here, a much larger surface here. And so, which box is going to have a higher rate of flux? Well, the one with the higher surface area. The more surface area there is, the higher the flux. This is why you have microvilli. This is why you have so many folds in your intestines to increase the surface area. This is why you have so many alveoli, and the structure of the alveoli in your lungs are the way they are. It's because it increases surface area. The more surface area you have, the faster molecules can move across the membrane. So if you want to have fast movement across a membrane, increase your surface area. Now let's look at this example. These orange hash marks have a higher resistance than the black hash marks. Which membrane, which box is going to have a higher rate of flux? Well, higher resistance in the orange membrane, lower resistance in the black membrane, you're going to have a higher rate of flux in the B box. It's going to be easier for those blue particles to move. Now, what affects membrane resistance in a human, in a cell? Well, the lipid and protein characteristics of the membrane. If you have a lot of cholesterol in your cell membrane, it's going to add resistance. If you have a lot of proteins in your cell membrane, it's also going to add resistance. Also particle size and solubility. So the larger the size, the more resistance. The more hydrophobic, the more lipophilic, the higher the solubility, the easier it's going to move across the membrane, the lower the resistance. Membrane thickness. So in this example, we have box A with a thick membrane and box B with a thin membrane. Which one is going to have a higher rate of flux? Well, thinner, less thickness in box B, there's going to be a higher rate of flux in box B. So let's put that all together. When you put this all together, it's called Fick's law. Fick's law summarizes the influences on direct membrane diffusion. So proportional variables are on the top of the equation, and by directly proportional variables are on top of the equation. Inversely proportional variables are on the bottom of the equation. A directly proportional variable means if you increase the concentration, you increase flux. If you increase the surface area, you increase flux. So these two are directly proportional. Inversely proportional means if you increase that variable, it decreases the outcome. So if you increase resistance, you decrease flux. If you increase membrane thickness, you decrease flux. And this only applies to direct diffusion particles across the membrane. So I'm going to give you a hypothetical right now. If you double the concentration gradient and keep everything else the same, what happens to net flux? If you put a 2 in front of concentration gradient, if you double concentration gradient, what happens to net flux? You put a 2 right here. Well, you're going to double flux. What I want you to do is I want you to put an imaginary 1 in front of each of these variables. Imaginary 1 in front of each one of these variables. And then if I ask you, double concentration gradient, how does net flux change? Well, that imaginary 1 becomes a 2 because you've doubled it. And you leave your 1 in front of the other terms. So we have 2 times 1 divided by 1 times 1. So 2 over 1 is equal to 2. You double the concentration gradient. Now, let's put our imaginary 1s back in front of each of these variables. And what happens if I double membrane thickness? So if I double membrane thickness, so how are we going to do this? So we have our imaginary 1s in front of every variable, but we put a 2 in front of membrane thickness for doubling of membrane thickness. Now we do math. 1 times 1 is equal to 1, so our top variable is 1. Underneath we have 1 times 2, so 2, so 1/2. If you double membrane thickness, flux will be 1/2 of what it used to be. You see how I do those equations? I have and I will ask questions like that on checkups and on tests. You just have to put that imaginary 1 in front of all your variables. And if you triple surface area, you turn that imaginary 1 to a 3. If you triple membrane resistance, you put that imaginary 3 in front of membrane resistance. Here's a good one. What if you double concentration gradient, but you also double membrane thickness? So you put your 2 in front of concentration gradient, imaginary 1 in front of surface area, imaginary 1 in front of membrane resistance, 2 in front of membrane thickness, because you double membrane thickness, you double concentration gradient. What happens to the flux? Well, 2 times 1, you've got a 2 on top. 1 times 2, you've got a 2 on the bottom. Flux stays the same. Because 2 over 2, divide that, you've got 1, and that flux stays the same. Channel proteins provide an aqueous pore for diffusion of lipophobic particles. So how are we going to get those ions into our cells? And not just ions, other things like water is another thing that goes through channels. So channels are net movement of light particles through a channel from an area of low concentration to high concentration to low concentration. Movement from high concentration to low concentration. So channels are tunnels. They're tubes. You can actually look at these on electron microscopes and you see a tunnel, you see a space where the particle moves right through. So channels allow for rapid transport of small particles. Rapid transport of small lipophobic water hydrophilic particles like water and ions are going to flow through these channels. Now, as we talked about in part 1, some channels have no gates. Some channels have 1 gate, they're single gated, and some channels have 2 gates, they're double gated. And there's lots of different things that can activate or open up these channels, like a ligand, something binding to the channel can open the gate. But reference part 1 for all the different parts, all the different types. Flux is going to depend on the concentration difference of particles, so concentration gradient, just like when we talked about Fick's law. But remember, Fick's law is only through the membrane. It doesn't count if you are talking about channels. And flux is also going to depend on these molecules that are moving through channels, on the number of channels. So the more channels you have, the faster these particles can move through those channels. Channels cannot be saturated physiologically. So what this means is there's no, in a human situation, increasing the concentration will always increase the rate of movement through these channels. There's no limit to the speed at which things can move through these channels. In a petri dish in a lab, yes, you can reach a limit to the number of particles that can move through. But in a human situation, you never reach a situation where there's a limit, a limit to the rate at which particles move through the channels. All right. So here it is. Single-gated channels and double-gated channels, and here are all the different things that might open or close a gate. So yeah, some channels are always open. They're less common. Most channels have some sort of gate and some sort of trigger to open or close that gate. We'll talk about double-gated channels when we talk about nerves a lot. We'll talk about all types of channels when we talk about nerves, but double-gated are really important when we talk about nerves. All right, so I'm going to stop part 2 here, and part 3 will pick up where we left off right here.